Hello everyone, it's Jihad Ali from Grand Canyon University, and the course is Concept of Human Physiology, Bio 515 or 500, and the presentation is all about endocrinology. So without further ado, let's get started. The posterior pituitary doesn't actually produce any hormones. It simply stores and on appropriate stimulation release into the blood two small peptide hormones, Vasopressin and Oxytocin, which are sensitized by neuronal cells body in the hypothalamus part of the brain. These hydrophilic peptides are made in both the supraoptic and paraventricular nuclei in the hypothalamus, but a single neuron can produce only one of these hormones. The synthesized hormones are packaged in secretory vesicles that are transported by motor proteins down the cytoplasm of the axon and stored in the neuronal terminal within the posterior pituitary. Each terminal stores either vasopressin or oxytocin. Thus, these hormones can be released independently as needed. On stimulatory input to the hypothalamus, either vasopressin or oxytocin is released into the systemic blood from the posterior pituitary by exocytosis of the appropriate secretory granules. Vasopressin, which is called antidiuretic hormones ADH, has two major roles effects that correspond to its two names. It conserves H2O during urine formations by kidney nephron as antidiuretic effect, and the other effect is cause contraction of the arterial smooth muscles, which is a vasopressor effect. The first effect has more physiological importance than the second one. Under normal condition, vasopressin is the primary endocrine factor that regulates urinary H2O loss and overall water balance. In contrast, typical levels of vasopressin play only a minor role in regulating blood pressure by means of promoting arterial vasoconstriction. The major control for the hypothalamic induced release of vasopressin from the posterior pituitary is input from the hypothalamic osmoreceptor, which increases vasopressin secretion in response to a rise in plasma osmolarity. A less powerful input from the left arterial volume receptors increase vasopressin secretion in response to a fall in extracellular fluid volume and arterial blood pressure. Oxytocin, on the other hand, stimulates contraction of uterine smooth muscles to help expel the infant during childbirth, and to promote ejections of milk from mammary gland breast during breastfeeding. Appropriately, oxytocin secretion is increased by reflexes that originate within the birth canals during childbirth and by reflexes that are triggered when the infant suckles the breast. In addition to those two major psychological effects, oxytocin influences a variety of behavior, especially maternal behavior. For example, the hormones also facilitate bonding or attachment between a mother and her infant. For this reason, oxytocin is sometimes nicknamed the love hormone. Thyroid hormone actually represents the combination of two main hormones that our thyroid gland releases, Thyroxine T4 and Tri-iodothyronine T3. They are often collectively referred to as a thyroid hormone. Because T4 is largely inactive, meaning it doesn't impact our cells, whereas T3 is active. Once our thyroid releases T4, certain organs in our body transform it into T3 so that it can impact our cells and our metabolism. The production and release of thyroid hormone, thyroxine T4 and Tri-iodothyronine T3 is controlled by feedback loop system that involve the following: hypothalamus, pituitary gland, thyroid gland, and multiple hormones. Hypothalamus is part of our brain that control function like blood pressure, heart rate, body temperature, and digestion, while pituitary gland is a small pea-sized gland located at the base of our brain below our hypothalamus. It makes and releases 8 hormones. Pituitary gland is connected to hypothalamus through a stack of blood vessels and nerve. This is called the pituitary stack. Through the stack, hypothalamus communicates with pituitary gland and tell us to release certain hormones. At the start of the feedback loop, hypothalamus releases thyroid releasing hormone TRH, which in turn stimulates pituitary gland to produce and release thyroid stimulating hormone. Thyroid-stimulating hormone then triggered our thyroid to produce T4 and T3 hormones. Of the total amount of hormones that thyroid-stimulating hormone trigger our thyroid to release, about 80% T4 and 20% is T3. Thyroid also needs adequate amount of iodine, a substance we get from the food that we eat, to create T4 and T3 hormone. This hormone chain reaction is regulated by a feedback loop so that when the level of T3 and T4 increases, they prevent the release of thyroid releasing hormone, and in addition to that, a thyroid stimulating hormone. When T3 and T4 levels drop, the feedback loop starts again. This system allows our body to maintain a constant level of thyroid hormone in our body. If there is any issue with our hypothalamus, pituitary gland, or thyroid gland, it can result in imbalance in the hormones involved in the system, including T3 and T4. Once thyroid releases thyroxine T4 into our bloodstream, certain cells in our body transform it into Tri-iodothyronine T3 through a process called deiodination. This is because cells that have receptors that receive the effect of thyroid hormone are better able to use T3 than T4. Therefore, T4 is generally considered to be the inactive form of thyroid hormone, and T3 is considered the active form of it. Cells in the following tissue, gland, organ, and body system can convert T4 to T3, which are liver, kidney, muscles, thyroid, pituitary gland, and brown adipose fat tissue. This type of fat produces heat to help maintain our body temperature in cold condition, and the other one is central nervous system. The thyroid hormone T3 and T4 affect every cells and all the organs in our body by regulating the rate at which our body uses calories, energy. This affects weight loss or weight gain, and this is called the metabolic rate. Slowing down or speeding up our heart rate, raising or lowering our body temperature, influencing the speed at which food moves through our digestive tract, affecting brain development, controlling the way our muscles contract, managing skin and bone maintenance by controlling the rate at which our body replaces dying cells. The anterior pituitary produces ACTH, which is Adrenocorticotropic hormone. It's considered a tropic hormone. As we know, the tropic hormone indirectly affects target cells by first stimulating other endocrine glands. Corticotropin-Releasing hormone CRH is released from the hypothalamus, which in turn stimulates the anterior pituitary to release adrenocorticotropic hormone. ACTH then acts on its target organ, the adrenal cortex. The adrenal cortex secretes glucocorticoids, a hormone primarily cortisol from the zona fasciculatus, and androgen from the zone reticularis. The secretion of glucocorticoid provides a negative feedback loop for inhibiting the release of CRH and ACTH from the hypothalamus and pituitary. Respectively, stress stimulates the release of adrenocorticotropic hormone. As we know, this is like a tool, like the memory tool for secretory product for the adrenal cortex. The deeper we go, the sweeter we get. So, for example, the glomerulosa release salt, mineral corticoid hormone. Fasciculatus reduce sugar, cortisol, and reticularis release 6 hormones, androgen. The adrenal cortex produces a great variety of hormones that any other steroidogenic tissue. Slightly variation in structure confer different functional capability on the various adrenocortical hormones, which can be divided into 3 categories based on their primary actions. First one is mineral corticoid, mainly aldosterone, and influences mainly minerals electrolyte balance, specifically sodium and potassium balance. The second one is the glucocorticoid, primarily cortisol, play a major role in glucose metabolism as well as in protein and lipid metabolism and in adaptation to stress. The third one is sex hormone, are identical or similar to those produced by gonads, tests in males and ovaries in females. The most abundant and physiological important of the adrenocortical 6 hormone is dihydroepianosterone, an androgen or male 6 hormone. So let's talk about the insulin and glucagon. Glucagon and insulin are both important hormones that play an essential role in regulating blood glucose, sugar. Both hormones come from pancreas. Alpha cells in our pancreas make and release a glucagon, and beta cells in our pancreas make and release insulin. The difference is in how these hormones contribute to blood sugar regulations. Glucagon increases blood sugar levels, whereas insulin decreases blood sugar levels. If our pancreas doesn't make enough insulin, or our body doesn't use it properly, we can have high blood sugar, hyperglycemia, which lead to diabetes. Glucagon, on the other hand, and glucagon are not the same. Glucagon is stored form of glucose sugar. Our body primarily stores glucagon in our liver and muscles. Glucagon is a hormone that triggers liver glucagon to convert back into glucose and enter our bloodstream so that our body can use it for energy. Parathyroid hormone is a peptide hormone secreted by parathyroid gland that regulates the serum calcium concentration through its effect on bone, kidney, and intestine. It influences bone remodeling, which is an ongoing process in which bone tissue is alternately resorbed and rebuilt over time. Parathyroid hormone is secreted in response to low blood serum calcium levels. Parathyroid hormone indirectly stimulates osteoclast activity within the bone matrix in an effort to release more ionic calcium into the blood to elevate a low serum calcium level. The bone acts as a bank of calcium from which the body can make withdrawal as needed to keep the amount of calcium in the blood at the appropriate level despite the ever-present challenge of metabolism, stress, and nutritional variations. Parathyroid hormone is a key that unlocks the embank vault to remove the calcium. In bone, parathyroid hormone enhances the release of calcium from the large reservoir contained in the bones. Bone resorption is the normal destruction of bone by osteoclasts, which are indirectly stimulated by parathyroid hormone. Stimulation is indirect since osteoclasts do not have receptors for parathyroid hormone. Rather, parathyroid hormone binds to osteoblasts, the cell responsible for creating bone. Via the kidney, parathyroid hormone enhances the absorption of calcium in the intestine by increasing the production of activated vitamin D. Vitamin D activation occurs in the kidney. Parathyroid hormone regulates 25-hydroxy-vitamin D3 1-alpha hydroxylase, the enzyme responsible for the alpha-1 hydroxylation of 25-hydroxy-vitamin D, converting vitamin D to its active form. This activated form of vitamin D increases the absorption of calcium by the intestine via calbindin. Parathyroid hormone only influences the resorption of calcium ions that occur in distal tubules and the renal collecting ducts. A more important effect of parathyroid hormone on the kidney is, however, its inhibition of reabsorption of phosphate from the tubular fluid, resulting in decrease in the plasma phosphate concentration. Phosphate ions form water-insoluble salt within the calcium. Thus, a decrease in the phosphate concentration of the blood plasma increases the amount of calcium that is ionized. A third important effect of parathyroid on the kidney is its stimulation of the conversion of 25-hydroxy-vitamin D into 1,25-dihydroxy-vitamin D, which is released into the circulation. This form of vitamin D is the active hormone which stimulates calcium uptake from the intestine. Let's talk about the calcitonin, which is another hormone, also called thyrocalcitonin, a protein hormone synthesized and secreted in humans and other mammals, primarily by parafollicular cells, C cells in the thyroid gland. The hormone participated in calcium and phosphorus metabolism in many ways. Calcitonin counteracts parathyroid hormone actions and vitamin D. More specifically, calcitonin lowers blood calcium levels in two ways. Major effect inhibit the osteoclast activity in bones which break down the bone, and another minor effect inhibit the renotubular reabsorption of calcium ion and phosphate, allowing them to be accepted in the urine. A growth hormone, also called somatotropin or human growth hormone, is a peptide hormone secreted by the anterior lobe of the pituitary gland. It stimulates the growth of essentially all tissue of the body, including bone. Growth hormone is sensitized and secreted by anterior pituitary cells, as I mentioned, called somatotrophs, which release between 1 and 2 million milligrams of the hormone each day. Growth hormone is vital for normal physical growth in children. Its levels rise progressively during childhood and peak during the growth that occurs in puberty. In biochemical terms, growth hormones stimulate protein synthesis and increase fat breakdown to provide the energy necessary for tissue growth. It also antagonizes the action of insulin. A growth hormone may act directly on tissue, but much of its effect is mediated by stimulation of liver and other tissue to produce and release insulin-like growth factor, primarily insulin-like growth factor, IGF-1, and formerly called somatomedin. The term insulin-like growth factor is derived from the ability of high concentrations of these factors to mimic the action of insulin, although their primary action to stimulate a growth. Serum IGF-1 concentration increases progressively with age in children, with an accelerated increase at time of a pubertal growth spurt. After puberty, the concentration of insulin growth factor 1 gradually decreases with ages, as do growth hormone concentration. Growth hormone secretion is stimulated by growth hormone releasing hormone and inhibited by somatostatin. In addition, growth hormone secretion is pulsatile, with surge in secretion occurring after the onset of deep sleep that are especially prominent at the time of puberty. In normal subjects, growth hormone secretion increases in response to decreased food intake and to physiological stress, and decrease in response to food ingestion. However, some individuals are affected by abnormality in growth hormone secretions, which involve either deficiency or overabundance of this hormone. Follicle stimulating hormone FSH, one of the two gonadotropic hormones. Hormone concerned with the regulation of the activity of the gonad or sex glands, produced by the pituitary gland. Follicle stimulating hormone is a glycoprotein operating in conjunction with luteinizing hormone LH. Stimulate development of the graphene follicle, a small egg containing vesicle in the ovary of the female mammal. In the male, it promotes the development of the tubules of the testes and the differentiation of the sperm. Though in the male, the presence of the follicle stimulating hormone is necessary for the maturation of the spermatozoa. Additional follicle stimulating hormone may not be required for months because testosterone can maintain this activity. In the female, however, there is a rhythmic or cyclic increase and decrease of follicle stimulating hormone, which is essential for monthly ovulations. Luteinizing hormone, on the other hand, is another gonadotropin, and it is a glycoprotein with a molecular weight of 26,000 in human. In the female mammal, it promotes the transformation following release of the egg ovulation of the graphene follicle into the corpus luteum, which is an endocrine gland. In the male, luteinizing hormone promotes the development of the interstitial tissue leading cells of the testes and hence promote secretion of the male sex hormone, testosterone. It may be associated with follicle stimulating hormone as I mentioned earlier in this function. The interrelationship of luteinizing hormone and follicle stimulating hormone has made it difficult to establish with certainty that two separate hormones exist, particularly since both are glycoproteins. Let's talk about prolactin, also called luteotropic hormones. Is a protein hormone produced by the pituitary gland of the mammals that act with other hormones to initiate the secretion of the milk by the mammary glands. On the evolutionary scales, prolactin is an ancient hormone serving multiple roles in mediating the care of a progeny, sometimes called the parent hormone. It is a large protein molecule that is synthesized and is secreted from lactotrophs, which constitute about 20% of the anterior pituitary gland and are located largely in the lateral region of the gland. In women, the major action of prolactin is to initiate and sustain lactations. In breastfeeding mothers, tactile stimulation of the nipples and the breast by the suckling infant block the secretion of the hypothalamic dopamine, which normally inhibits prolactin, into the hypophyseal portal circulation of the pituitary gland. This results in a sharp rise in serum prolactin concentration, followed by abrupt fall when feeding stops. High serum prolactin concentration inhibits secretion of gonadotropin-releasing hormone from the hypothalamus, thereby decreasing the secretion of the gonadotropin, luteinizing hormone, and follicle stimulating hormone, and may also inhibit the action of gonadotropin on the gonads. Thus, high serum prolactin concentration during lactation reduces fertility, protecting lactating women from a premature pregnancy. And finally, here are my references. Thank you for listening, and God bless you all.